Hi, my name is Justin Stewart, and I'm going to be talking about how we need to get back to basics. There are ecological questions in microbial ecology that we can answer, but we aren't. This format's going to be a little different than a normal Inspire talk because this whole year is different, and this is what I want to do, and so I'm doing it. So 16S studies are very, very helpful. They're cheap, they're easy to do, but they miss a lot of biodiversity. A lot of new methods have come out recently. Specifically, we can look at the three-dimensional configuration of chromosomes and be able to look at the rare biosphere. Furthermore, we're also able to use metagenomics to disassemble and look at the functional and taxonomic diversity of these microorganisms. And recently, some phylogenetic models have come out that are, have been applied to studies of the human microbiome that could also be applied to different environmental samples to help figure out these questions that I'm about to present to you. So keep in mind that there are a lot of really unique ways that we can do this that we just aren't. So the goal of all of this is to inform and direct future research programs and agendas and essentially just benefit humankind and human flourishing. What was that? Eudaimonia from Aristotle. So we're looking forward to that. The first thing that comes to my mind that we are not doing is we are not forecasting change, and we are not predicting and quantifying uncertainty in these predictions of how ecosystems and the services they provide are going to change in the future. So a quick Google search for the keywords of microbial ecology and forecasting just came with 12 results over the last years, and I constrained this to the last 10 years because I don't think we really have like the computational abilities to predict things very well. And this is something that I think we really need to do to create a more sustainable future. Things that we need to keep in mind with this are that things are spatially and temporally dependent. So if you look right here, the closer you are to things, like the scale that you're looking at is going to be really important. So that's something to keep in mind. Like if you were over here in Italy, you might focus on different scales of, like, let's say this is wind, than if you were looking at the whole Mediterranean or chaparral area over here. The other thing is to look at temporal scales. Things at shorter time scales are harder to forecast because we don't see as much seasonality in them as we would at longer temporal time scales. In figuring out how to solve short-term iterative forecasting is something that's really important in microbial ecology and that we need to all be working on. Not all, whoever wants to work on it. I love your work. Some things to keep in mind with this are the fact that data with forecasted data is autocorrelated. So there's a law in geography, Tobler's law. Everything is related to everything else, but near things are more related to distant things. This thing normally holds true if you're living in a city. You're going to go and walk about and it's going to pretty much look like a city, but it can also fall apart. Let's say you're walking in nature and then you walk up to a cliff. There's a very large change in elevation in a very short time. So with these models and with our data, we need to be correcting for these autocorrelations spatially as well as temporally. So things that occur more closely together in time are more similar. Surface temperature of a city is more similar around noon to 12:30 than it's going to be from noon to 8 p.m. because the sun will have gone down depending on where you are longitudinally and the time of year. Next thing is we need to talk about urban ecology. There are bacteria in the city. Cities are the fastest growing urban environments. We need to be understanding how these microbiomes are interacting with human populations that live there to create more sustainable cities. Another really interesting thing that I've been thinking about is that cities are not homogeneous. Cities are heterogeneous in their structure. Rich people like to live in areas that have a lot more green space because who wouldn't want to live there? But it disenfranchises different populations. So it can disenfranchise minorities and BIPOC. And we know that this structure holds inequality. Are these communities poorly served with their microbial services is a question that I think we also need to be answering. Microbial ecology does contain social justice, and I think that we all are able to incorporate that. Specifically at this time, we all need to be bringing that into our research. There are, again, as I kind of mentioned, some difficulties again with this. Urban areas are extremely heterogeneous. So there's grass, there's trees, there's pavement, there's low-rise buildings, mid-rise buildings. And looking at this three-dimensional configuration is really difficult for trying to understand microbial communities in a city and what they're actually doing for us. The next big thing is to look at global patterns. Are microbes in Tempe in Arizona the same as the microbes you're going to find in Guangzhou in China? And what are these structures in these cities that we can find redundant patterns, and how can we utilize these patterns to make cities more sustainable and better for humanity and the Earth, obviously. Space, oh my gosh, we have satellites. We can also do remote sensing of microbial diversity. So very few studies have actually looked at this. There's one from Larson in 2014, where they used remote sensing combined with truth samples to essentially look at functional gene content. And it's really fascinating because they are able to, and we are able to, use satellites to capture and interpolate continuous biodiversity. Here they did functional traits, which I very much value. And this is incredibly powerful because, one, it's essentially free data. Well, I mean, like, you know, we're paying for it for taxes, but it's free data that's generated recurring, and we're going to have historical records. So matching up microbial sampling with some of these remote sensing days may actually help create better models to figure out how to search for, say, extraterrestrial life, or to make, again, a more sustainable environment for us to live in. Again, difficulties with that. There is some paucity in the data. So for example, here's Landsat 8, or a rendition of it. It goes over every 8 days. And so you would have to sample, like, you know, every 8 days or in those 8-day intervals. And at the same time, you have to be aware of the weather conditions. You know, there might be a cloud covering where you're looking at. And it of course depends on what you're looking at. So if you're looking at like NDVI or biomass of plants, that versus surface temperature are going to be impacted differently based off of weather conditions. The other thing is it can be computationally expensive to deal with all of these rasters. So we need to find ways to leverage advances in computer science to actually marry these two, microbes and satellites, together, which sounds crazy, right? Like satellites and microbes, what? Resilience. Okay, so we know that ecological resilience is when there has been a disturbance, either a long-term or a short-term one in a community. And when I say community, I mean either taxonomic structure, phylogenetic, or functional. And then we're talking about the return to that original state. So I'm not talking about resistance right here, but one thing we need to look at is how are these communities resilient to disturbance? And this, again, I'm kind of focusing in on urban areas because that's one of my favorite things. And how can we engineer resilience into these either urban areas or natural environments so that we are able to, under global change scenarios, exploit these properties to increase human flourishing and make sure, you know, we don't die. So again, there are urban applications of this. So I immediately thought of like the High Line in New York City. How can we exploit that to? How can we exploit the resilience of these microbial communities that are constantly exposed to stressors like particulate matter in the atmosphere, humans walking on them, that Instagram influencer sitting down, taking the photos, crushing the microbes on the phylosphere? And then also it's really helpful under global change scenarios to be able to figure out how can we make sure that a community will be able to still function and maintain the functions that we need and produce ecosystem services while the environment is actually changing. Humanity. So these are a little more, a little different, and these are kind of a call to action. We really need to figure out how we can supply microbial information to policymakers. What is the role of environmental justice in microbial systems? And that kind of goes back again to talking about like could microbes in an urban environment be structured in a racist or disenfranchising way? And then how can we replace microbial fear with excitement? How many of you guys have talked about the fact that you're a microbiologist or a microbial ecologist, and people are like, ew, or they immediately ask about pathogens, but then ignore like fungus, literally moving all the carbon. So these are some of the questions that we ask. And honestly, these need to be done through outreach and scientists taking elected offices. So I'm not saying you have to. I'm saying this is something I think that we need to think about more often when we're going through school. The next thing is there are other ways you can do this without actually having to be in, you know, Congress or whatever your governing body is. You can definitely use social media. Younger people like me have been doing interesting things. I've seen people stream their research on Twitch, which is used for like video gaming. People do live tweet things on it. They go live on their Instagram stories. And I think it's helping normalize discussing microbial systems and destigmatizing the idea of all microbes being pathogens to the general population, or at least depending on the cohort of people you follow. And I think we needed to expand that, because if we're doing science and we're not talking about it, then in my opinion, you're not doing science. which may be a controversial opinion, but I'm controversial. And then also, I think we could actually have interventions at younger stages with for children and teach them about microbial systems. Take them out, show them how we can grow a microbe on a petri dish, or explain, you know, what's going on in the rhizosphere in different ways. What are the nodules in a legume? Something like that. I think there are definitely ways that we are able to answer questions to the general public that we aren't actually exploiting. So this was a pretty short talk. I mean, it's inspiring. I only did 15 slides because there's a pandemic and I'm like moving. So yeah. If you have any questions, feel free to DM me. My Twitter is @thecrobe, like the microbe. This has been a lot of fun. I look forward to seeing all of your talks.